and look and see if it looks right. So.
Careful, you're talking to someone who's failed photography at least twice, but I remember.
I suppose we can look at, specimens or samples when they're on the deck, but who's to say that the color isn't different in air versus in 1,000 meters of water?
Let's come down to 5, Jake, and see what happens to. Is Nicole still back there poking the DSC, is she?
Yeah.
What are we gonna do down here? We're gonna collect some specimens, transects.
All of the above. Let me know when you're settled and we'll chat about it.
Yeah, we can chat. I think we have a few people working in the background, but we're almost there.
Okay. We will be running some transects as usual, like we did yesterday, especially now that we're in hard habitat. It'll be a good one to do. We will be collecting a whole coral colony at some point. I wanted to check with Taylor Ann. It may be too soon in the dive to do that. It will ooze a little bit in the bio box. And we will want to collect a Niskin about 50 meters above it. So we may do that later, but we don't want to lose out an opportunity to not see it again. So maybe Taylor Ann can provide some advice on if we were to collect it now or later.
Yeah, I'm not too sure what the difference would be. We have everything open right now. You could cut it up and put it into a forward bio box. Those are the biggest boxes that we have available. But yeah, I guess if you're cutting it into pieces, you just want to be careful that you're not losing any of it. But yeah, it's up to you guys if you want to explore around and see. I'm sure, like, I don't know, there might be more of these around. There seems to be a lot in this area.
Yeah, once we're done with the camera, we might also get a zoom out and see what's around. There are a couple species here.
If you look in Argus right now, you get a good idea of what's around there. It's
yeah,
bit of a forest.
So Jan mentioned we didn't want ones with sweeper tentacles, but there are some keratoisis specimens here that don't have that are also requested. So we'll take a look around once we're done with the camera setup.
Sounds good.
Beautiful shot, especially with Argus.
Is that her done, is it? Pull up the DSC, would you?
Yep.
Oh, we got a bottom gauge reading, did we?
Just seeing what Nicole said on the way out. Oh, you didn't hear? Okay.
Oh.
No.
Should I stop it and?
Yeah, let's do it.
All right. Cancel.
Cancel or stop up there. I think you have to hit stop and move that out of the way. Okay. Steve, if you zoom out on both cameras now, we'll come up and have a look around. You don't have to do all the focus stuff. Just hit start and stop again.
Yeah, we're
up. That guy. Move that, move that dialog box out of the way first. Yeah.
Oh,
Okay, so I got all that. We're gonna look around. Is that right?
Yeah, we could just take a look around where we are right now, see how many corals are around. We may hold off on collecting a sample. We'll see.
Good to go there?
good to go.
Got it.
So about the white balance, I think the ideal situation would be to get that white further out into the light that Herc is emitting.
Oh, we got lots of stuff here.
You want to put Bubble back in the front for me?
Yep.
Bunch of those stationary holothurians that I forget the name of.
Solus.
Solus.
Zoom in just a touch for us there, Steve. Yeah, thanks. What are we looking for again, or we're just looking?
We're just looking. Taylor Ann, if you could make a notation that there are both keratoisis and isodella in this area.
Got it.
And Taylor Ann, just a note that when we do bring up one of these bamboos tonight, it will be slimy and you'll probably want to wash it out on the deck on a tarp.
Yeah, I see.
Before you bring it to the lab.
Jan's notes there.
Okay.
Yeah.
Oh, good.
I'll pass those on to the data loggers after me as well.
Okay. Okay. I'm hoping we're going to see a few more of these. I would suggest we see it sort of orient ourselves towards moving towards the next waypoint from here.
Roger that. As you circle around, Dan, explore. Our eventual heading will be 065.
Roger.
Wow, some really interesting sponges.
Yeah, this is hard bottom habitat, so we could start a transect at any time. Whatever feels good to you all on board.
We could start it right off the bat. I think it's pretty, it's not too steep of a slope on the way towards 2K.
That'd be nice, and we'll just make our way towards 2K, yeah.
Sure.
So let us know when you're ready.
Roger.
Bit of a change. We land on the cool stuff and not deliver it to the next watch.
Just watch that. Hold what you got there, Jake, and watch that tether as they come around. Just hold what you got there, but.
Yep.
And that means don't do anything.
Oh. I thought you meant come up, get out of the way.
Come back down.
Okay.
I want to see the tether. Bob's got me paranoid about it. You can, yeah, look down a little with your camera now.
Let me know when you've started.
Yeah, we haven't started yet. We're just getting sorted out here.
Great.
I will let you know.
What was the bearing?
065.
Roger. Look down some, Jake, with your camera.
That's about as.
Is that all the way down?
Yeah, I'm like full down.
All right, the current's got it then.
I can turn more if you want.
Yeah, try that. Whatever you gotta do to, I want to see it, what's happening. Should be all right with this current. That'll help us to.
So to interrupt, Jan would like us to get some good photos before we leave this area for a transect.
Sounds good.
So it looks like we got some open space here to settle down. Is that true?
Yeah, I'll have to turn and put my nose into the current there. So
Looks like
you
that's the
get
way
good
that,
photos.
yeah, that's the way the coral are facing anyway, so it'll be a good.
We're pull up the Jake.
Yeah.
Pull up the DSC on that. You want to get what kind of photos are you wanting? Sorry.
We could get some zooms on a couple of these corals would be nice, if you're close enough. There's several species in the area here.
Okay, yeah. It's a little weird because I gotta use it for the mouse and stuff too, but. Yeah.
I'm assuming Jan would like some photos with lasers and then some glamour shots without them, without the lasers.
Roger, I'm also setting up for landing here. We have a really nice digital stills camera on the vehicle at the moment, so you should get some high resolution digital still photos.
Great.
Is that position of that camera set in stone, or is it possible?
Yeah, it's hard. It's hard mounted at 45. Well, we get a preview of what's happening here with it. So, we also have the Jake's deep eye.
Yep. But can you change that stills camera between dives?
The deep eye one is more easily.
Give us a bump to the north for any, please.
Roger. Bridge nav. Step 15 meters north.
Come down 3 meters, Jake.
Roger.
Make it 4.
4.
Okay, can zoom in there.
Mostly closed polyps on this one at the moment.
Do you want to keep the lasers in for now?
Yes.
I'm not sure what they want to.
Yes, that'd be good. And I like to see what's living on it. So we do have some anemones and some hydroids with some bare skeleton on the left. So those are starting to eat the coral polyps. Can we see the whole animal? Zoom out a little bit.
What?
Yeah, great. So this is Keratoisis. It's a bamboo coral. I think all three in that view are the same species.
You can come up a couple of meters on Argus now. Cool shot though.
Yeah, and maybe turn the lasers off for a couple glamour shots would be great.
Yeah, great
Off.
image.
That's great.
Yeah.
And then if we could perhaps zoom in at the top where those anemones are feeding.
Can do. Okay, Steve.
That's great right there. Thank you. So we often see other animals climbing up on these bamboo corals, either to getting a better space in the current, but also they're feeding on the polyps. We got a photobomber back there, a grenadier.
Total photo bomb.
I have a little more zoom here if we want to get even tighter.
Sure,
Yeah.
just to
And that's it.
it's a nice illustration of some of the coral polyps are out and some are in. All right, that's great. Thank you.
It's interesting that one branch on the left, it's got all its polyps are in except for one.
The last holdout. He's hungry. Jan's asking what kind of anemone, but I do not know what kind of anemone that is.
Okay,
And
moving
without
on.
without moving the ROV, can you turn to the left a little bit to see what's on the left, or is that difficult to do?
Yeah, we want. You can. You won't have real good light over there.
Nope, just like that. I just wanted to see what was there. Okay, so that one on the left is a different species. That's Isidella, and it does have the sweeper tentacles, but these again are all bamboo corals in the same family.
Can zoom in there a bit, Steve. Maybe get a better shot. So.
Sure, it might not be perfect, but sure, give it a try.
Is this the one we're going for, or the one further left?
It's further left, I think.
Would you like the lasers
Sure.
back on?
Yeah.
But yeah.
Okay, off we go. You want to look at the
Yes,
one
thank
on the
you.
left closer? Do you? I can scoot over there, or we can find another one.
I think we're good. If we can
Okay.
try and get back on our transect idea, that'd be great. I think we got enough shots there of those. Thank you.
Sounds good. Once you get set up on 065, we'll call the start of the transect. Big one.
God, they're just monsters.
Yeah. Doing well down here. This clear viz is really really spectacular.
Just gonna pause here and get a let our digital stills cam takes a shot every 30 seconds. This will get really good pictures and for sitting on the seabed because then the object's not moving. So.
One more interruption. Jan would like to take a look at that sponge straight ahead, that big white trumpet sponge.
Me too.
Okay, good. I would like to entertain you as much as we can, so it's
Thank
not just
you.
boring for you guys.
Should we redo the white balance on the sponge?
Yeah, that would be a white balance. We should do it. Do a save as. What kind of sponges did you call it? Sorry.
I call it the trumpet sponge.
Right.
This one is sort of spilled over, but it
It's
sometimes
massive.
looks taller. Yeah. And I think the genus is Conalasma. But sponges are so difficult to identify sometimes, but I think that's what it may be.
Come down 5,
And
please.
so that all that sediment in the middle means that the sponge is not clearing its cells very well in there. So it may begin to die, or maybe it can eventually push that off, but that is a clogging problem.
You want to bump over there? Or are you good? We'll get a good auger shot from here if you're happy with the tether.
Yeah, happy. I'm just going to back up a bit and try and get that in our stills camera. You'll be able to see that down inside there a little better. Zoom in there if you want, Steve.
Full zoom there.
Yeah, I'm not sure what they want to see. Probably the whole thing.
Full body shot.
Full body without the lasers would be cool.
Roger.
This is a glass sponge, and to me at depth it always looks so soft and inviting, but they can be pretty prickly with their glass spicules. Great, thank you. Were you going to do a white balance, or were you just kidding?
No, I was just joking.
We're just
Okay.
bantering.
Okay. All right, I think we're ready. See if Jan pipes in again.
Erica, what are these small stick branching?
Go zoom in one more
Yeah,
time,
so-
Steve.
Sure.
Yeah,
she's
to
asking
those-
about that.
Those sticks, those are the predatory sponge Asbestopluma. And instead of filtering, they have Velcro-like structures that can actually capture their prey and make them immobile. And then they start to digest it with their, I don't know if it's an enzyme or what.
Wow.
There was a request for Asbestopluma. Let me check to see if we got it.
Did we get one
did on the
yesterday?
last dive.
Yeah.
Okay, that's right. Gary collected a couple, I think. Okay, we can move on then.
Yeah.
Okay, Steve, zoom out, please. Thank you.
Or any head. See you throw any heads. So we don't often get to see quite such a forest of bamboo corals, and so that's why we're taking so much time, kind of just getting some great pictures and capturing this place before we start our standardized transect.
Yeah, and as the ROV pilots mentioned, there's a strong current here right now. And I would imagine the size of these corals, they've been here a while, so that current might persist because they like to be in the current. So this is a good place for them.
Jake, you want to go over to 065?
Yes.
Okay.
Ready for the ship move?
Yeah.
Okay, we'll hold off on starting the transect until we see Argus move, but sorry, where's my bridge nav? We move the ship 150 meters, bearing 065, speed 0.5.
Let's try start out at 0.3.
Bridge.
Oh no, sorry, yeah, 0.5 is fine.
Okay. Thank you. And this will be H1824 T1B, is that correct?
Oh, shark.
Yes.
What species is that?
988 meters.
It might be another cat shark.
Zoom in just a little bit for us, Steve.
Coming in.
It's a
Yep,
very
that's good.
pretty nose. Yeah. Beautiful.
Check, check. Check, 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 check.
Did we get a sense for size on that?
Can you confirm you have the lasers on?
Lasers.
We've had good luck with cat sharks, haven't we?
I don't quite have the leash to get a close-up of him yet.
Yeah, getting spun.
It's bouncing at the end of my leash here. Can try zooming in a bit, Steve. Bouncy zoom. Zoom a little more. More. Okay.
About 40 centimeters, maybe.
Yeah, little guy.
I'm amazed it's sticking around so long. Okay, that was cool.
Just waiting for the ship to start moving. We're waiting for Argus to start moving. I should be able to go back to 065 there.
Roger.
See if our autos are going to play nice. So.
Those cat sharks, they lay egg cases, and occasionally we'll see egg cases attached to the base of corals and rocks. So we'll have to keep our eye out, see if we see them.
Okay, you guys ready?
Ready.
Ready.
What is that number? Okay, begin transect. Hey, got any more down on the Argus camera?
A lot of those predatory sponges in here. Some of the sessile cucumbers, the solus, the white branching. A mushroom coral down on the right, just leaving the screen. Few thorny head rockfish.
Coming up across some more bamboo coral. We've got Isidella. Those are the ones with the, looks like a stalk.
The
Sort of
ones
a
on the floor.
candelabra.
Wait, what do you say? Oh, okay.
Bridge now.
There's several.
Was that a
You can add
Swifty
to this
simplex?
50 meters, bearing 065. Thank you.
Yes, it was.
Are you getting any background sound on me?
Yes.
Sorry, dog.
So Swiftia is in the center. It's a little bush-like coral. And more Isidella. We just passed Keratoisis, another bamboo. And there's a dead skeleton laying on the seafloor. We're just about passing it lower center. That happens sometimes. If they don't adhere to a strong enough rock, they can easily fall over if the current picks up too much. Nice habitat. Thanks for dialing it in, Dan and Lenny.
So that seems
Yeah,
right.
we're just driving the bus. You guys picked it. Awesome job.
That seems like then that's meters per second. This seems wrong. It's anybody's guess. Yeah. Yeah, but that doesn't even seem right. That should be 0.5. Yeah, it could be some nav cook actually, or the navist. Hmm. Mm-hmm. Yep.
The substrate is really interesting. Pebbly and
Mm-hmm.
all those different colors too.
Yeah, it sort of looks like broken rock, but I guess there's cobble in there too, huh?
Yeah, a little of both.
Yeah, we're pretty consistently on the ship 0.58, so that'll account for that. All right, that's 100 meters, so we'll continue on to 150,
Yes,
unless you want longer than that.
150 is good. We're getting some really good data here, so I think that'll be great.
Great.
Can we just keep moving and go on and off transect, or how do you want to do that?
I think at the end of this transect, we might want to do some collections. And Jan has requested a Niskin be triggered.
Yeah.
Yeah, copy.
So
I can trigger the Niskin on the flat.
sure.
Sitting here with my hands in my pockets right now.
Yeah, it's pretty undisturbed seafloor. Well, you know, that'd be great. Just tell us when you trigger it.
Yeah, I'll wait till after the transect, but it's,
Okay, perfect.
We will need to get that space at least 100 meters between transects anyway.
Right.
Mm-hmm. It does. But sample collection. Yeah.
Erica, the two that are ahead of us right now, at least the one on the right, is that dead?
I don't
Or
think so. I think the
polyps?
polyps are in.
I thought these
Yeah, let's
don't
see.
aren't really able to
About another
move in
25.
alone.
Yeah, I guess I'd have to get a close-up view, but we won't do that now.
Right.
And that actually may be Cibogorgia.
Yeah.
So that's why I'm thinking at the end of this, we hopefully will see some more of those and we'll collect it. So I've seen several so far already.
Do we want to drop? Oh, okay.
You want to, sorry, what was that? You want to drop a waypoint there?
Sure, just say drop a waypoint and name it Cibogorgia.
Oh, that's easy.
Do your best. Starts with an S.
S-Gorgia. That's what you get.
That's fine. We'll know what it is. But I'm hoping we'll see some more. I'm seeing some more right now in front of us. So by the end of this transect, I think we'll have some.
Okay. Something Gorgia. What was it again? Just one more time.
Cibogorgia.
Can you use that in a sentence,
Sure.
please?
S-Gorgia.
For how many points?
Okay, the ship has stopped its move, so I can add another there if you want, if that's what you're saying, but we're going to change course to 08. Okay.
This glass.
You still have a few more meters to go. Bridge nav.
That felt fast. Was it just that we had so much to look at?
Still want to go half a knot? Let's do a ship move 50 meters, bearing 088, speed 0.3. Okay, that's the end of transect there. We got,
Great.
Yep, just bang on 150 meters. So.
Okay. That's great.
Niskin?
Niskin?
Please.
Yeah, right.
Did you say 50 meters above?
No, let's do it here. This is good.
Okay.
That was it, wasn't it?
Looks like it.
Looks like, yeah.
And what was the sample number on that one, Taylor?
That was 15. So 015, 015.
Rog.
You slowed him to 0.3, did you?
Roger.
Thank you.
I think your Argus is still going in that 065, but eventually we'll change to 088.
To 0 what?
088.
088, roger.
All right. I think I see some corals up ahead. So we're going to look for that Cibogorgia. And then we may also collect a bamboo, which for aging, which does require us to take a Niskin 50 meters above the bottom. Is that hard to do at this point in the dive?
Oh, it's easy to do.
Okay. So let me look at our data sheet here.
So if you see a likely suspect, let us know, because we're still moving the ship
I
and
will.
the ROVs, plural.
Okay. I'm looking.
We're less likely to tangle that tether up if we keep everything moving. Is kind of where I'm at with that.
May I
Yeah.
interrupt?
Yep.
There's a coral right at the bottom center. This one right in view.
Okay.
That
Charlie
looks like
Brown
a good
coral?
one to collect.
You'd like to collect it?
The bushy one right. We're coming out of frame now at the center bottom. You're just passing it.
Was that a collect?
Yes, that's a
Could
record.
be.
Bridge nav. Let's hold position here.
With reference to the lasers?
I think you're on top of it, so it's beneath you.
It's in the, yep,
Right there.
that one, bottom left.
Right to the left. Yep.
Big one or the little one?
The big one. Well, it's big-ish. Yes, the big one. Right at the bottom center view.
Copy.
Sort of orange. Yep. So Taylor Ann, we can break it up. Will that fit in the bio box?
I think Dan would know more than I would. Can you get that in the bio
I'm
box?
sorry.
Oh yeah, stuff a Christmas tree in that bio box.
And we would like the whole thing, including the base, which can be hard to pluck off. So if the rock is small enough, you can collect the whole rock, but if not, you can get as much as the base as you can.
There's one tipped over on a rock.
Yeah, we don't want that one.
Species. Starfish on top of them.
Yeah, I think it should
Bridge
fit
nav.
if we're able to cut it up in a few pieces, maybe. But we don't want to, I don't know. Yeah, I think it would be okay.
20 meters, 315.
Just gonna, before we collect it, I'm gonna set down momentarily and we'll get a digital still of it.
Great.
We'll get some zooms from here as well. Okay, Steve. I'll do a laser
8
zoom
feet from,
first.
are we more than 20 feet
That's
from the
good.
last Niskin bottle?
I will measure that. Give me a moment. We are 20 meters from the last Niskin.
Okay. So we will, Jan is requesting, once we collect this, or now, we'll want another Niskin at depth and then 50 meters above it.
You go on a Niskin here on the seabed and the one above, is that correct?
Yes, yes, please.
Copy.
Do you want to lose the lasers at this point, Dan?
It's up to the scientists, but I imagine they'll want a laser less shot as well.
Yes, please.
Killing lasers.
And what is the name of this coral? Is this the bamboo coral
This
we
is
saw earlier? The keratosis?
Yes, this is, yes, keratoisis.
Okay, thank you.
And a zoom would be good on the polyps.
Coming in?
Yeah, freeze it.
Great, thank you. Jan is asking for some 360 photos of this. Is that hard to lift up enough?
No, it's easy. photo
Okay.
mosaic. My co-pilot has some Flash software to make a model too.
Use Colmap?
Yeah.
Yeah.
Coming wide.
Roger.
Now, Dan, let me know when you think I'm at a good zoom here for this.
And let's try that.
Sure.
I'll probably have to adjust because I need to come up a bit. Otherwise, I'll knock everything down in all radius. I don't want to do that. Just gonna play with the auto here for a second and get better pictures if the computer's flying. You might have to step a bit closer.
I called a 315 20 meters. We're just starting to feel that, inching closer to you now.
Roger.
But let me know if you want more.
And this is the sample that you would like to be rinsed prior to entering the lab, correct? That's what we said.
Yeah,
earlier?
this one can ooze a little bit. The other bamboo is much slimier, but this one as well can emit a bunch of mucus. So I guess it's better to remove the mucus for aging studies. And it's cleaner in the lab too. It gets kind of messy. So in this view, we do see some fallen over corals, and that sometimes happens when they get too tall or the rocks below them are too small to hold them up.
So are we starting this, are you clicking a bunch back there, Taylor Ann?
What was that?
Are we clicking a bunch for this photo mosaic on the
Oh,
Herc
yeah.
HD? Okay.
Yeah,
Just
all
double
the
checking.
click.
Should I increase the frame rate on the DSC too, or get some?
No.
No, just leave it.
Yeah.
All right.
Let it do its thing. You have to stop the auto shooting and start another folder and blah, blah, blah.
You might notice in the background that fallen over Isodella has a bunch of sea stars and the Tritonia nudibranch on it. So they may have arrived after it fell, or they might have made it top heavy and it fell over. But the coral itself looks pretty clean, so I imagine they found it after it fell over. I mean, it's full of polyps, I should say. It's not cleaned up yet. But they're feeding pretty well on that thing. So this 360 helps the taxonomist piece back together the coral because it will likely break up when we sample it. So they can reconstruct it by looking at these images. To know what branch went where, it'll help with the aging part of the study. This little eel pout curled up, sort of floating by. I think that's great. I think you can collect it whenever you have a chance.
I'm gonna go ahead and come around the other side here. We didn't do 360 yet.
Oh, I'm sorry.
Came around halfway and then came back, and then we'll come around halfway this way and come back. Still paranoid about putting a turn in that tether.
I think that's a good paranoia. So what's Bob think there's already one in it?
And they had a wrap on recovery.
Yeah.
So in that case, we lay it out all on the deck and we look at it and talk about it and trying to decide if we want to take the wrap out once we're subsea again, which is what we did there when we first came down. And so far, I'm thinking that it looks okay. If we wind up with a hockle halfway through the dive, I will really regret that decision. So the other thing is to put one in or take one out and see how it looks, but that can, we'd be there for like half an hour at least playing around. Really hard to justify when you're in a coral forest.
Yeah.
So being conservative with the we also want it to last for the rest of the season. So it's about 20 hours to manufacture another one. We of course have 2 spares in the box, but
Gotta connect them.
yeah, it's not too hard to put another one on. It's just making the replacement.
Yeah.
Cost of the tether and man hours, parts. Usually we get one last the whole season. Okay, I think that was pretty much a double 360. It was a 180, 180.
Great. Thank you.
I'm hoping we get
With
some
the
great
rock?
high resolution stills. If you're lucky, we
With
can
that
just
rock?
pick up that rock and
Exactly. That would
put
be
the whole
great if
thing in
it's
the
small
box.
enough. Perfect.
Okay, you want to?
Well, Taylor Ann,
I need to just get a little closer here.
Taylor Ann, what sample would this be? What number?
This would be sample number 16.
Thank you.
Can you? Oh, never mind. Never mind.